Our colleague, Dr. Pritha Goswami, Assistant Professor, Geology Department, will take us through the significance of systematic paleontology as a foundation for paleoecological studies. Madam, are you present?
Yes, sir. Sir, I will be presenting my screen now. Please let me know that if it is visible. Is it visible, sir?
Yes, your slides are visible. Please go for the full
Uh,
screen mode.
yes.
Yes, it is
So,
visible.
thank you, sir, for your introduction. Good afternoon, everybody. Uh, I would like to discuss with you today the topic systematic paleontology as the base of paleoecological studies, a case study from the fossil record. So, as we all know, that systematics is the study of the diversity of organisms and the interrelation among them. And a major part of the systematics is basically taxonomy, where we are naming these organisms, we are describing these organisms. And when coming to taxonomy, this taxonomy leads us to various branches of paleontology. So when we are naming these fossils, we are taken into the science of paleobiogeography, paleoecology, biostratigraphy, and all these. So let's try to understand that when we are naming a particular fossil, then that indicates a certain geologic range. And the presence of that fossil in the rock signifies that the rock was deposited during that time. So that brings us to the subject of biostratigraphy. Then, when we count the tree rings or the annual growth rings of the corals, we are taken to the subject of geochronometry, which gives us the time through which our annual cycle of the Earth prevailed. So, data like this take us to give us the information that our Earth is slowing down in its pace. This idea is also backed by the geophysical data, and we get to know a lot about the celestial phenomenon from this geochronometry. Taxonomy is very much related to the functional aspects, and therefore it also post-Darwinism. It encompasses the idea of evolutionary theory. So information like evolutionary theory, the growth and the forms, the different functional morphologies which speak about the ecology, the habitat, everything is obtained from the clear and accurate taxonomic studies. Apart from this, when we are studying about different organisms, their presence at certain localities, like a fossil is present in, suppose, India and Africa, a terrestrial animal, and they are now actually miles apart, separated by the oceans. So we can just go back into time and think about this and try to understand that might be there was a time when these two land masses were together. So that gives us the idea of paleobiogeography and plate tectonics as a large, at large. And paleoecology, as we all know, it is the interaction between the organisms and the organism and the environment. And so these kind of studies are also initiated from the proper taxonomic studies. So when we come to the definition of species, we know that the biological definition of species, that is a biospecies concept, does not hold good in case of the paleontologic species that we are working with. We generally work with the morphospecies, where we depend solely on the morphological aspects to name the species. We cannot make these animals participate in the experiments, the breeding experiments. We cannot see their biochemistry and all because these are already rocks that we are working with. So morphology is the only thing which we need to decipher, and we need to minutely decipher that, keeping in mind the evolutionary context. So in order to do that, what I did was that we collected specimens from different localities within Kutch, Gujarat. And this is the entire Cenozoic stratigraphy of Kutch. And I have worked with the Khadi Nodi formation and the Chhastra formation, which ages back to 23 to 15 million annum ago. And these two formations are basically having these rock types and are largely fossiliferous. I have selected 10 localities from where, within these two formations, from where we have collected the samples. And we got about 25,766 complete and near complete gastropods, from which we documented 51 species within these two formations. And as you all can see that these gastropods, these are the taritelids, as many of us know. So this assemblage was particularly a taritella dominated community. And with the proper examination of the morphological traits, we have named this species and we have grouped them into different genus, and then they have been grouped into families and so on. So let's try to appreciate the beautiful morphologies that these gastropods are having, that we have unearthed. So after unearthing these gastropods, naming them, what we wanted to know was whether this diversity was adequate, like whether we have collected adequate fossils to document the near biological truth of the place. So rarefaction was the method which could be implemented in this process, and therefore this is an ideal rarefaction curve. If I try to make you understand that if we go on increasing the number of the samples, the number of the species would be increasing until this curve attains a plateau, which indicates that further increasing the number of the samples won't be increasing the species. Therefore, the true biological diversity of that place has been reached. So when I run this analysis in the lower Miocene level, as well as the two formation levels that I have worked with, we saw that the peak was trying to attain, and therefore the biological diversity of Miocene time was perhaps collected. Thereafter, we tried to compare the diversity between these two formations, and we saw that there was an overlapping range of diversity within those 8 million years time. As I have collected samples from the spatially different locations, so we wanted to find out whether there was any spatial heterogeneity in the diversity values of the fossil record. And we saw that it was also more or less overlapping with some of the places showing a bit of anomaly. And then after this documentation of diversity, we moved on to the different paleoecological studies. So as prey-predator interaction goes on in the recent scenario, as it's evident from this video, you can see here that a bivalve and gastropod are in a predatory activity where this gastropod and here another gastropod are involved in this predatory activity, and such kind of predatory activities leave back a trace which is also preserved in the fossil record, and that is known as the drill holes. So these kind of drill holes having these kind of geometries are preserved in the fossil record, and we looked for the prey-predator interaction in the fossil record after proper documentation of the diversity. So we saw that there were a number of species and there was good predation pressure also over there. And we wanted to know that whether the abundance of the species was somehow related to the drilling intensity, that is the predation pressure. On examining that in both the formations, we saw that there was no relation between abundance and drilling intensity. So the most abundant gastropods were really not the most drilled ones. So after that, what we did was that we compared our data, our Miocene predation intensity data to the global record. And we saw that within the Miocene high of drilling intensities, our data was really very low. So we also wanted to know that is the reason behind such low drilling intensity in the Miocene of Kutch. So what we did next was that we studied the paleobiogeography. That is, we took the map of lower Miocene of the world. As you all can see, this map is very similar to the map of today. But obviously, it has certain seaways opened and closed, which are very different from our recent world. So this is a lower Miocene map, and we see that this Kutch falls under the Western Indian province. We divided the entire Earth into different paleobiogeographic provinces. And then we saw that this Kutch diversity had a very high endemicity, 62.18% endemicity of the species. And we could understand from that that perhaps the endemic forms that we get in Kutch, they were naive, they were inexperienced, and they were less escalated to be good predators, resulting in the low drilling intensity. So such kind of beautiful studies and interactions can be very well documented only and only if we correctly identify the species. If we do this taxonomic study with much care, because in case of our paleontology, we are only using the morphospecies, and therefore the variations and the species both should be very well dealt of, and then they can be implemented in different paleoecological studies. So this is all I wanted to tell. These are my major references, and thank you all for your patient hearing.
Thank you, madam.
Thank
The
you
floor
so
is
much.
open for questions.
I suppose there were no questions at opposite side.
Then, madam, I have one query. Just
Yes,
as you were
please,
doing
please,
those
please.
rarefaction curves, these are very similar
Yes,
for our biodiversity analysis in case of
yes,
plants or
yes, yes,
insects.
of
So,
course.
is there
Yes.
anything for diversity analysis of these gastropods?
Similar curves.
Yes, I mean similar indices can be used, or there are something else.
Yes, yes, there are like we use the taxonomic richness, the species richness. We use the some coefficients to check whether, for the two formations, whether the diversity is significantly different or not. These kind of statistical analysis we do with these also.
Okay, madam. Okay, you got one question from Priyankar Chakraborty. This is in the chat box.
Okay,
How do
I'll
you
just
link the fossilized animals to their present day counterparts?
Yes. So thank you for the question. It is really very interesting. In geology at large, we say that present is the key to the past, with obviously certain exceptions because the earth is ever changing and the dynamics is also ever changing. So the thing is that I have publications in the recent record also of gastropod and barnacles and all. So the thing is that we study these recent counterparts to understand that how they are behaving among themselves, how they are interacting with the environment. Because in the past, we cannot go to the past and see these things for real. So our laboratory is the fossil record, and in case of paleontology, what I'm working with, vertebrates and invertebrates, I generally visit the oceans. I work with marine fossils generally. So I visit the sea beaches and all intertidal region, and I get to see a lot of animals, present day animals, the gastropods and bivalves. The movie I showed you was in one of our field work only at Chandipur, Orissa, and we see that how they are associated with nature. Like we have bivalves which are shallow in faunal. So who are shallow in faunal, their commissure line are often infested with barnacles. So the presence of barnacle on a bivalve shell in suppose the anterior or the posterior side often signifies that that barnacle was infested in the live like live assemblage only. Like it showed that it was a shallow marine fauna. So such kind of comparison we can easily do from the recent counterparts to the fossil animals. Am I clear, Priyankar? Okay, thank you so much.
Thank you, madam.
Thank you, sir.
You just stop sharing and we will move to the next presentation. Okay. The next one we have Vishal M. Makwana. He will be presenting on agonistic behavior of red-wattled lapwings under anthropogenic pressure. Are you present?
Yes, sir.
Yes, sir.